Thank you very much. Glad to be here with you today. Go through a quick presentation about the University of Florida peanut varieties that are available for 2014 and beyond. There are 3 relatively new high oleic peanut cultivars that we've released in the past several years. I won't mention Varieties 7, that one's been out there quite a while, and the seed supply of all these is around 2,500 to 3,500 acres of certified seed produced in 2013. So just to give you a rough idea of how much is out there, I'll show you a slide at the end that gives you an idea of the total amount of high oleic seed out there. But the Flora 107 was released in 2010, Tough Runner 727 in 2012, and the Tough Runner 511 was released just last July. So the Flora 107 is a high-yielding cultivar with good that one either. We'll be developing that over the next couple of years. We talked a lot about high oleic peanuts, and just wanted to give you an idea of why this is important to the industry. This is a comparison of a high oleic variety here and a non-high oleic here, and something called peroxide value, which is a chemical measure of how the peanuts would taste to you after they've been stored for a while. And a peroxide value of about 20 is when the peanuts start to taste rancid and you begin to notice that off flavor. When they compared a high oleic peanut here in the dark line to a normal oleic, a high oleic peanut here in this line at the bottom to a normal oleic here over many weeks of storage here, up to 36 weeks of storage, what they found was that in 4 weeks they began to notice this off flavor in the normal oleic peanut, whereas it took 32 weeks to notice that same off flavor in a high oleic. So this is why the high oleic is so important to the industry. In this particular preparation of peanuts, this was in-shell roasted. So that's one of the preparations that becomes rancid the most rapid. So this is why the high oleic peanut is important to the industry. I mentioned the seed acreage a minute ago. If you look at the whole acreage of certified seed in Alabama, Florida, and Georgia, about 79% of it is still in Georgia 06G as of 2013. And I know some of you are interested in the high oleic contracts. If you look at the amount of high oleic seed available, about 10% of that in total is made up of the Florida 07, Tough Runner 727, Flora 107, and Georgia 09B. Those are the 4 high oleics that are there, so only about 10% of the acreage in high oleic seed from 2013. And also did something similar, a little more quick and dirty than what Dr. Smith showed earlier. This is a comparison of the value per acre given the contract that was issued for $4.25 a ton for normal oleic and $4.75 a ton for high oleic. I was curious with that how that would make the varieties compare. So I compared the 4 high oleics here to Georgia 06G, obviously the dominant cultivar on the market today. And if you look just quickly at the irrigated in Midville, the irrigated in Plains, and the irrigated in Tifton, you can see that adding that $50 a ton premium really makes those varieties, all the high oleics, more attractive from an economic standpoint, at least in Tifton and in Midville, not so much in the Plains data. And this is 3 years of data from the Georgia variety test. So just gives you an idea of the competitive nature given that contract price difference. Similar to what Dr. Smith showed earlier, that under irrigation, the high oleics look economically quite attractive at those price differentials. With that, I'll close and just invite you to our field day we'll have in August in Mariana and ask for any questions.
Good morning. I'd like to just spend a few minutes talking about 2 things. First, we'll start with a little bit of discussion on Tipguard. Most of you are aware of Tipguard, so we're not going to spend a lot of time on it. And then the second part of my talk is going to be talking about the next variety that's in the pipeline that should be released shortly. Seed won't be available, but I just wanted to go ahead and tell you about that. So to begin with Tipguard, as most of you probably know, Tipguard is a high-yielding, medium maturity cultivar with high resistance to the peanut root knot nematode, high resistance to the
tomato spotted wilt virus, and moderate resistance to leaf spot. This is a picture of a test that Dr. Tim Brenneman had a couple of years ago. The 3 treatments were susceptible variety with no nematicide, a susceptible variety with nematicide, and Tipguard without nematicide. And the plot you see over here on your left is the Tipguard without nematicide. It was a field with very heavy nematode pressure. This is a bit of a further away shot, but you could, just stand on the edge of the field and pick out the Tipguard plots without any nematicide. Any of you that have a problem with the Rootknot nematode are probably aware of all the information on this slide. The juvenile female establishes a feeding site in the roots of the peanut, and that causes the formation of a giant cell, which causes the galling on the roots. Within a fairly short period of time, like about 30 days, 30 to 35 days, she produces 300 to 500 eggs, which are deposited in an egg sac, and they, stick out from the roots. Those eggs hatch, and then all those juveniles go and infect nearby peanut roots. So with heavy infestation, of course, you got bad damage on the roots and even bad damage on the pegs in the pods. And the nematologists estimate that approximately 25% of the peanut fields in the southeast have damaging levels of the peanut Rootknot nematode. A little bit of data from that test that I showed you with Dr. Tim Brenneman. The susceptible variety with no nematicide, a lot of galling, reduced yield. The nematicide treatment was 10 pounds of Timic at planting and 10 pounds at mid-bloom. And it did reduce the galling and increase the yield. But the best treatment in this test was Tipguard with no nematicide, significantly higher yield than the other treatments. A test that we conducted in another field, these were all cultivars with no nematicides in a field with nematode pressure. And once again, your best option would be Tipguard. So now I want to move on to the thing that's coming next. And it's just known by a number right now, C1805-3-43. This release package has been submitted to the official Variety Release Committee, along with a whole lot of data. And next year we will have a name that I can share with you, but right now there's no seed available, so it's just known by a number. The objective of this work was to develop a nematode-resistant variety with high Oleic. So we began by crossing Tipguard, which provided the gene for nematode resistance, with Florida 07, which provided the genes for high Oleic. This work was in close cooperation with Dr. Peggy Ozias-Aiken's group. She's a molecular geneticist and has molecular markers for both these traits. This is some of the data that we get from her on the nematode-resistant trait. It's very powerful technology. We can get this, we can take a little leaf tissue from an individual peanut plant, and she can tell us if it's homozygous recessive, which would be homozygous resistant, homozygous susceptible, and she can even tell us the individuals that are heterozygous. We get the same information for the high Oleic characteristic. In this case, this is 76 different plant samples that we gave to her lab. And in this case, each one of these lines is an individual plant. And these are the plants that were homozygous for high Oleic. The red plants are homozygous for normal Oleic. And the blue plants have one allele that's high Oleic and one allele that's normal Oleic. And this is really powerful tool that the conventional breeders can now use. You probably have heard about the genome sequencing effort, and I think Darlene's going to tell us a little bit more about that later. But we hope through that genome sequencing effort, which is an international effort, that we will have this type of technology for many more traits, like resistance to white mold, resistance to leaf spot. And I just want to take this opportunity to kind of set Darlene up. A little bit of data. I don't want to provide a lot of data, but I did want to show you how this line is holding up. This was the first yield test that we had back in 2010. That's another breeding line of ours, but you can see yield per acre for this line was very good in comparison to the other cultivars in the test. This is a test that Dr. Tim Brenneman conducts every year. It's called the Multi-State Disease Evaluation Test, and he opens this up to all the breeders, so you can give him seed of your breeding lines, and he'll test it in this test. It's a very severe test, very little fungicides for foliar diseases, no fungicides for soil-borne diseases, and he inoculates 6 plants in each plot with white mold. So the yields are not ever very high, but the relative yield for this line was excellent in comparison to the cultivar checks. We work with Dr. Brenneman and do a greenhouse screen on that set of genotypes that he tests every year. And this is the greenhouse screen for resistance to the Rootknot nematode. And you can see this line is similar to Tipguard. And these are the susceptible checks. You can just focus here. This is a mean of 3 different tests in 2011. Once again, 1805-3-43 performed very good in comparison to other standard checks. This is a test that Dr. Albert Culbreth conducts every year, and he plants it early at a low seeding rate to try and get severe tomato spotted wilt pressure. He had severe tomato spotted wilt pressure in 2011, and so you can see, of course, Georgia Green had a lot of tomato spotted wilt with reduced yield. These 2 lines had better resistance with better yield. 1805-3-43 had the best resistance and excellent yield. We also had this in the Alabama State Variety Trials in 2012, and it performed very well. So in summary, this line is high yielding, nematode resistance, high Oleic, resistance to tomato spotted wilt. I didn't provide you with the data, but I did to the release committee. It also has a moderate level of resistance to leaf spot. I also provided data to the release committee. We did a bunch of hole scrape, and it is medium maturity. I'd like to conclude with the way I concluded last year, and just a couple of comments on the management of using nematode-resistant cultivars. This is a single dominant gene that was introgressed from a wild species. As you know, pathogens are very crafty at overcoming resistances, and so just because we have this high level of resistance, we still should follow good crop rotation so that we'll have this tool for as long as possible.